Again, there's issues about downstream from this, any nonsense-mediated decay. Felix Jimenez, who's here at this meeting, has been working with the rat and has had help from a technician in the lab. Now, when you look at exon 7 and 8, now in the null rat, I think that you see there's still considerable transcript downstream. The exon 6 is similar in being large and having many methionine codons that could reinitiate. And I guess if I was to make a new rat, I would delete all the exons or some very large number of the exons. We may do that because the current rat, interestingly, at the facility that made the animal, has them infected with Pneumocystis carinii, and they sent them to us that way, and we discovered that and informed them of that. So we're trying to rederive the existing rat to get rid of the Pneumocystis infection. So Rodney Samako is a faculty member in our department who does a lot more behavior than I do, so I've asked him to get involved in doing more in-depth behavior of the old Angelman mutation and the new Angelman mutation. And he's developed a plan here, which I won't go through the individual tests. This is partway through the analysis what some of the data show, and he's displayed it here such that a darker blue is a higher p-value. So you see that in some of the locomotion tests, there's higher p-values. You have, here's the new mutation up here, the old mutation down here, and you have males, females, and combined. So there's some hint, and we have a new locomotion, a foot slip test, which we think may have some advantages, so we may see some maybe better distinction of the old Angelman and the new Angelman mutation. Here in the marble burying test, which tends to be one of the more robust tests for these animals, it seems like the old mutation and the new mutation both have impairment in the marble burying test that's comparable to work published from other labs. And we're doing the fear conditioning. This is early times. I would say there's nothing conclusive at this time. And I must say, having watched fear conditioning attempt to be implemented in the Knockout Mouse project around the globe at like 20 different sites in 8 different countries, it's really hard to get these things to be extremely robust. And so Rodney has some people who've been working with him to do the behavior work. So some conclusions on the mouse behavior. It's very challenging. You really have to watch out for confounders like the weight of the mouse. There may be some lessening of the phenotype in the old exon 5 compared to the new exon 6 deletion. There's much more downstream transcript in the old exon 5 deletion than in the new exon 5 deletion. We have some things that are reacting with antibodies that we're not sure if they're real or not, smaller molecular weight things which we're trying to characterize by mass spec. So at this point, it's not clear if in the old mutation, it's certainly possible that there could be a protein made that has ubiquitin ligase activity. And I think the lesson going forward is if you really want to null, you'd probably better delete multiple exons, if not all the exons. So my overall, I would say let's see here. So I don't, that slide seems, anyway, that slide's out of place. So I've said that Ionis wants this humanized mouse. So this is what they envision, that you take a big chunk of the relevant region in the human and you slip it down in here in the mouse. You might or might not delete some mouse region. And now you would be able to take a human ASO and put it into a mouse, and it should activate the paternal copy of the mouse gene. And they feel like many of these ASOs behave very differently in vivo than they do in cell culture, and that they don't want to take an ASO, a human ASO, into the clinic without knowing about a good bit about toxicity study and its efficacy in an intact animal. So we've been mainly responsible for trying to help them with this, and I would say I greatly underestimated how difficult this would be, and we've lost time that has certainly slowed down the potential for any phase 1 trial. So you could insert small amounts, 50 kb, 50 base pairs or 100 base pairs quite easily with CRISPR. You probably can insert 5, 10, or 20 kb with CRISPR with some effort, but it gets increasingly difficult with increasing size. And these would be into the desired region. But Ionis has ideas of why not put it this backs. We've got an 85 kb back and a 184 kb back. Why can't we put the whole back in there? There's no precedent for putting human backs into mice in a location-specific and orientation-specific way. If you put things in a random orientation, you may have to include a promoter. So they are pursuing with a core that's quite experienced in making back transgenics, doing a non-specific integration of the back, but then you have to have a promoter at the right end of the back so that it will express antisense. And I would say also, we don't really know that is this going to be this simple. Maybe if you put a big chunk of human DNA into the mouse region, it won't behave the way you want it to behave. It may start forming loops and causing problems. They want something big because they want to screen lots and lots of ASOs, both for the ability to knock down antisense and for toxicity. So we do have a 20 kb fragment that's been introduced in a random location, not in a desired location, and we're trying to introduce a promoter at the proper end of the 20 kb insert. That should be fairly straightforward with CRISPR, and we should have something like that fairly quickly. We're trying to insert into this region. If you look at the mouse here, and we're looking at the end of UBE3A over here and SNO RNAs over here, you can see that there's a lot of inverted dupes and repetitive sequences through here. There's a little window here where there's no repetitive sequences or inverted dupes, and so this is where we're trying to insert the human material. Whether it would be good or bad to delete some of the mouse region, we don't know. These are the location of two of the backs they're interested in. The larger one includes a good bit of SNORD 115, as you can see here, and includes also the terminal portion of UBE3A. So this is human material that they would like to see inserted into the mouse. There are docking strategies. This is one that was published by a group at Stanford and has been commercialized by a company using the ATTB and other ATT integration mechanisms. So the company called Applied Stem Cell, they've kind of called this Target. You insert first the dock, that is this ATTB site, into some preferred part of the mouse genome, and we have actually contracted with these people to put this dock into the desired site. Then you come back with an integration system, and you have an ATTB site here in the material you want to insert. You have an ATTP site that you've now introduced into the mouse genome, and this works in such a way that you can integrate, but you can't come back out. And that's the advantage of the way the recombination sites are used. So this would be one docking strategy, which we're just initiating with this company. One can also make a similar docking strategy with inserting an a LOX-P and a LOX511, which is a mutant form of a LOX-P site, into the region, and we have been successful in putting this kind of a dock into the desired region. And Ming Zhu has been doing the bulk of the work with that. but he's had help from the mouse CRISPR core and had some help with constructs from another person that used to work in our lab. So let's see, I had an out of place slide here. So this is a few conclusions. Angelman is extremely favorable for the possibility of a cure by a number of sort of happenstance aspects, I think. The knockout mice can be complicated. I think there's always concern that what people think are null mice might not really be null. And that maybe even alleles can acquire suppression or other modifiers while they're being maintained, and there's precedent for this in other organisms. There may be modest differences between Angelman genotypes. We heard today that the deletions would have only one copy of this UBE3A modifier, whereas the other genotypes would have two copies of that modifier. I've said again and again that regulation of UBE3A may be very complex, and I think we've really moved getting the humanized mouse to the top level, although Stomy and I have talked at this meeting about trying to convince Ionis that maybe they can get the human oligos they need without having the humanized mouse, or at least have some alternative strategies to get there. So I'll stop at this point. There are people from Ionis are listed here. Frank Rigo in particular has been the main player on their end. And we've had funding from NIH and the Angelman Foundation. Okay, thank you.